Hello, hi everyone. I'm Sandhya. I'm your botany lecturer. In this video, we are going to study about the eukaryotic cell. As the name itself says eukaryotic cell. Karyo means nucleus. U means true nucleus. True nucleus in the sense the nucleus is covered by a membrane. Membrane bound nucleus is present. Double membrane bound nucleus is present. True nucleus. Advanced nucleus is present where the genetic material is having the chromosomes. It can be condensed to chromosomes. And these type of cells, these eukaryotic cells are present in protists, fungi, plant cells and animal cells. Plant cells and animal cells. Then it is having organized nucleus. Nuclear membrane is present. Nuclear membrane is present and the nucleus is called as organized nucleus. And it can be differentiated into, the genetic material can be differentiated into the chromosomes. Genetic material is organized into chromosomes. Then coming on to this particular eukaryotic cell, about the plant and animal cell. Okay, difference between plant cell and animal cell. Okay, difference between plant cell and animal cell. Main difference is that in plant cells the cell wall is present. Presence of cell wall. Here absence of cell wall. Absence of cell wall. Then here plastids are present. Plastids, that is chloroplast, all double membrane plastids are present. Here absence of plastids. Plastids are totally absent in animals. And here a large central vacuole will be present. Central vacuole will be present. Here the vacuoles are small vacuoles. Big vacuoles are not present. This is the general difference between plant cells and animal cells. And here no centrioles are present. Whereas in animal cells, the centrioles are present, which are useful for cell division, production of spindle fibers and like that, all those things. Centrioles are present. Okay, mainly centrioles are present. Okay. So in plant cell no centrioles, in animal cell centrioles are present. So if you see the structure of a plant cell, okay, like this it is going to be. A big central vacuole, the nucleus, here the chloroplast. The chloroplast will be present. Okay, this is called as cell membrane. This is the cell wall. This is the vacuole. This is the nucleus. This is the cytoplasm. Okay, this main thing, the plastid, the chloroplasts are present. In detail about plastids we will study. We'll study in the next class. Okay. Chloroplast. If you take about animal cell, here animal cell, if you take, there is no cell wall. Cell membrane is present. Centrally placed nucleus is present. Here the mitochondria is present. The centrioles are present. Ribosomes are present. Here the no chloroplast is present. Okay. Here just cell membrane. Cell membrane, nucleus, mitochondria. Mitochondria is also present in plant cell. Okay, centrioles are present. And other structures are present, but major difference is that cell wall is present in a plant cell, cell wall is absent. Plastids are present in plant cell, plastids are absent in animal cell. Large central vacuoles are present, but they are smaller. Centrioles are absent here, but animal cell centrioles are present. So this is a major difference between the plant cell and animal cell. Coming on to the general structure of a eukaryotic cell. I want to show it in the form of a flow chart. Okay, I want to show it in the form of a flow chart. Let us see now. Okay. So this eukaryotic is divided into plant cell and animal cell. Plant cell and animal cell. Okay. So in plant cell, cell wall is present, which is divided into, we'll study in detail about it. It is divided into middle lamella, primary cell wall, and secondary cell wall. Okay, whereas here it is having protoplast. Okay, it is having protoplast. Okay, it is having protoplast. Okay, this plant cell is also having protoplast and cell wall. Okay, this protoplast is nothing but cell membrane and protoplasm, isn't it? Protoplasm. Okay. So protoplasm is nothing but again cytoplasm and nucleus, isn't it? The cytoplasm is nothing but again cell organelles which will be floating in the cytosol. Cytosol, the water will be present, at the same time some reservoir materials, dissolved materials will be present in the cytosol. The nucleus is having again two again. It is nuclear membrane, then nucleoplasm. This nucleoplasm is again having the general nucleosol, and it is having nucleolus and the genetic material. Okay, the nucleolus and the genetic material, chromatin reticulum, or you can say it as chromatin reticulum. Okay, so in the cell organelles, it is again divided into endomembrane system, which is composing of endoplasmic reticulum, Golgi complex, lysosomes, vacuoles, mitochondria, plastids, then centrosomes. These are all membrane bound, and also they are also having non-membrane bound cell organelles such as ribosomes. Okay. Like this. Non-membrane bound, these are membrane bound, and few more are present. So this is about the general structural details, or you can say the parts of a eukaryotic cell. Okay, so plant cell and animal cell. In plant cell, cell wall will be present. This is absent in animal cells. So the plant cell is having cell wall and protoplast, whereas animal cell is having only protoplast. cell wall, middle lamella, primary cell wall, secondary cell wall. We'll study in detail. Protoplasm is nothing but the protoplasm plus cell membrane. The protoplasm is nothing but cytoplasm and nucleus. Cytoplasm is having cell organelles and cytosol. Cytosol is nothing but the water is present and some of the substances which are dissolved in the particular water. Cytosol. In the cytosol, the cell organelles will be floating, freely floating in the cytosol. Some are membrane bound, some are non-membrane bound. Non-membrane bound are ribosomes. Membrane bound, the endomembrane system is having endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. Mitochondria, plastids, centrosomes. Centrosomes are present like this. Coming on to nuclear membrane. Nuclear membrane is having same structure as that of cell membrane. Then and the cell membrane of the prokaryotes is also same. The cell membrane of the prokaryotes and the eukaryotes is same. And the nuclear membrane of the nucleus is also the same structural composition. Then the nuclear membrane is going to enclose the nucleoplasm. Nucleoplasm is going to have nucleolus, a darkly stained body nucleolus, which is useful for synthesis of rRNA. Then chromatin reticulum, which will get condensed into chromosomes. So this is about the general structural details of the eukaryotic cell. Now we are going to study in detail about the cell wall now. Okay, in detail about the cell wall. Cell wall was first discovered by Robert Hooke. How can you say that? That is same. Okay, cell wall. See, the first discovered cell was a dead cell. Dead cell. It was discovered in cork tissue, dead tissue. So the structure which he observed was the cell wall. So who observed? Robert Hooke. Robert Hooke observed the cell wall. It is a dead tissue. Okay, dead tissue. In the dead cell, we have the cork tissue. He has discovered it. It is the cell wall is going to give the structural support to the cell. It gives shape, and it also protects the cell from bursting or collapsing. Then this particular cell wall is present. It is present only in plant cells, not in animal cells. Then it gives shape to the cell. It is going to protect the cell from mechanical damage and infection, and it is also very much important for cell to cell interaction. And also it is also going to act as a barrier to the undesirable macromolecules. It is, or you can say it is a permeable membrane. It is a permeable membrane. Cell to cell interactions are going to take place, and also it is going to protect the cell from the undesirable macromolecules. In algae it is made up of cellulose, galactans, mannans, minerals like calcium carbonate, like that. But in plants it is made up of cellulose. In plants, cellulose, hemicellulose, pectin and proteins. It is made up of proteins. This is about the chemical composition of plant cell wall. Okay, plant cell wall. Coming on to the structure now. Structure. First, in a young cell, the primary wall will be formed. Primary cell wall is going to form in a young cell. What happens? Which is very much useful for the mechanical support. Then, as the cell matures, what happens? A secondary cell wall is going to be formed towards the cell membrane. Okay, Okay, this is the secondary cell wall in a matured cell. Secondary cell wall. This is the primary cell wall. Sometimes it's going to have some small gaps which are called as plasmodesmata. Connections from one cell to another cell. Okay, connection from one cell to another cell. The secondary cell is going to form formed by the addition of few materials like lignin, suberin. It is going to form inner to the inner. It is going to form inner to the cell membrane by the deposition of the lignin and suberin. A secondary cell wall is going to form. Then unthickened areas are called as plasmodesmata, which are useful for intercellular transport. Then here in the early stage, when during the cell division, a cell plate is going to form. That cell plate is going to form into a middle lamella like this. It is going to have middle lamella. This is the primary cell wall. Primary cell wall. This is the middle lamella. This is the first cell wall and middle lamella. This is going to form from cell plate. Cell plate during cell division. In a mature plant cell, it is the outermost layer of the cell wall. It is the outermost layer of the cell wall, and this particular thing, the whole cell wall is going to have a small connections called as plasmodesmata. Plasmodesmata. Okay. This middle lamella is having calcium pectate. Chemical composition of middle lamella is calcium pectate. This is about the cell wall, which is the outermost layer of the plant cell wall, and it is a permeable permeable layer. It is going to give mechanical support, structure and shape to the plant cell or to the cell. This is about cell wall. Coming on to the cell membrane, plasma membrane. Okay, coming on to cell membrane, which is also called as cell envelope or plasma lemma. Or it is also called as plasma membrane. So many terms are there, okay? The plasma membrane, plasma lemma, like this. It is a selectively permeable membrane. Selectively permeable membrane. Or you can call it as a semi-permeable membrane. So this is about the cell membrane. Different names of the cell membrane. Why it is called a selectively permeable membrane? The cell wall is permeable to all molecules. Whereas this particular cell membrane is somewhat intelligent. It is going to allow only the selective molecules, selective substances to enter into the cell membrane. It is selectively permeable membrane. Why it is selectively permeable membrane? We are going to study now. So in detail we are going to study now cell membrane. The cell membrane is the one which is going to cover. It is the one which is going to cover all the cellular contents of a cell. All the cellular contents of a cell. Then the chemical composition of cell membrane is it is having lipids and proteins. It is having lipids and proteins. Hence, it is called as lipoproteinaceous layer also. Lipoproteinaceous layer. These lipids are present in the form of bilipid layer. Bilipid layer, like a sandwich. Like this. Okay, bilipid layer. Okay, like a sandwich. And this particular lipids, what type of lipids are present? Phosphoglycerides are present. Phosphoglycerides are present. So then how are these lipids arranged? Arranged in a bilipid layer. Two layers they are arranged, like in the form of heads and tails. Heads and tails, like this. Heads, and these are the tails. Okay, these are the tails. This is one. These are the tails. Like this. These are nothing but the hydrophilic heads. These are nothing but hydrophilic heads. Hydrophilic hydrophilic head, or you can say polar head. Okay, these are the tails. These both are the hydrophobic tails. Hydrophobic tails, which are non-polar. Okay, so these heads are towards the outer side, towards the outer side of the cell. These tails are towards the inner side. So hydrophilic means they are somewhat liking. Philic means liking. Hydro means water. They are aqueous. They are aqueous. Aqueous. They are water liking heads. Aqueous. This is hydrophobic. Somewhat having the lipid content more. It's having hydrocarbons. So hydrophobic tails. They are non-polar. They are non-polar. This is about the bilipid layer, which is present in the form of like a sandwich. Then here, how are they arranged? It is lipids now. Coming on to proteins now. Proteins are embedded in this particular lipid layer. Maybe totally embedded or partially embedded, or just on the surface of the lipid layer. Okay, on the lipid layer. Let us study now this structure in detail. Okay, these are the heads. Okay, sometimes the protein may be totally embedded. Okay, totally embedded. Sometimes may be peripheral. Sometimes here a sugar moiety may be present. Sometimes what happens here, the cholesterol will be present, like cholesterol. Okay, this is the sugar. I'm sorry. This is the sugar one, which is branched or unbranched like this. Okay, this is the structure of these are the tails, hydrophobic tails. Okay, so what is this? This is a sugar. This is a peripheral protein, which is just floating on the bilipid layer. Peripheral protein. Okay, what is this? This is the cholesterol. Okay, this is integral protein, which is partially embedded. Which is totally embedded is, this one is integral protein, which is totally embedded inside the bilipid layer. Inside the bilipid layer. Okay, so this is about the structure of the cell membrane. And this cell membrane was like a quasi-fluid nature. It is having a quasi-fluid nature, which was first explained by Singer and Nicholson. Singer and Nicholson. And this particular model is called as fluid mosaic model of cell membrane. Fluid mosaic model. So this particular cell membrane is not stationary. It is going to float like this. Float. It is going to move. Okay, it is going to move one matter. According to this, the quasi-fluid nature of lipid enables the lateral movement of proteins within the overall bilayer. that this ability to move within the membrane is understood in the fluidity. Like this, it is going to move. It is a living tissue. The cell wall is a non-living tissue. It is a living tissue. It is going to move. It is going to move. Okay. This is about the cell membrane. This is about the cell membrane, about the structure, and chemical composition. The fluid nature, because of the fluid nature, quasi-fluid nature of the cell membrane, it plays so many important functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. And also this particular cell membrane is useful for the transport of the food materials or transport of the substances in and out of the cell. The transport of the substances without the requirement of energy is called as passive transport. Okay, so this cell membrane is useful for passive transport. It is useful for diffusion. Diffusion means the movement of the substances from higher concentration to lower concentration. Then osmosis, the movement of the water from lower concentration to higher concentration through a semi-permeable membrane. The cell membrane is a semi-permeable membrane, osmosis. And also the movement of some solute particles with the help of the proteins or carrier proteins or ATP involvement. Energy requirement is important. Such type of transport is called as where the transport of the substances or solutes where the requirement of energy is must. Such type of transport is called as active transport. So these are the functions of the cell membrane. Thank you.